wither. So that's a play on wither biophysics, whereas biophysics don't. So single molecule functional analysis in the age of big data. Okay, so here's a paper that just came out. I think it's still an ASAP. It doesn't even have a citation yet. So this is from the Derizi lab, as probably many of you know here. Identification and characterization of a previously undescribed family of sequence-specific DNA binding domains. So this paper was published in 2013. I'm just going to really rip through sort of some highlights of this paper to start the talk off. So basically, they discovered a new DNA binding protein in C. albicans. So Candida albicans is the #1 fungal pathogen that infects and kills humans. People with AIDS, like a huge percentage of people with immunocompromised bodies, die from things like opportunistic infections. C. albicans is a big one. And so what's kind of interesting about C. albicans is it exists in 2 populations that are genetically identical but have a different phenotype. Basically, the colonies just look opaque or white, so you can see them with your eye. And so that colony phenotype that you can see, there's actually a lot of genetic regulation that's underlying that, but in 2013 it's still not exactly well understood why that's happening or everything that's controlled in that process. So because this organism causes human disease, this is an important thing to know about it, because this opaque, white and opaque thing also relates to the ability of the albicans colonies to mate. Okay, so basically what they did is they did RNA-seq profiling of white and opaque colonies and compared them. So then they also, so there were a couple proteins in this pathway that were known, proteins that are known to transcriptionally regulate this switching between the two. So switching is very rare, but it does occur. And you see upregulation and downregulation of this pathway depending on which state the particular colony happens to be in. And so by doing ChIP-seq against WR1, which is a transcription factor that is involved in this pathway, they were able to identify, by comparing that to the RNA-seq data that was already published in the literature, they found one gene, ORF19467, which was upregulated in the opaque cells, and by ChIP-seq they found that WR1 did bind upstream of this gene, meaning WR1 transcriptionally activated whatever this gene is. And so they call it WR3, and so by using Derizi's Myong 2.0, they found that it was indeed a sequence-specific transcription factor. So in 2013, we found a completely new class of DNA binding proteins that are sequence-specific. So the question is really like, how did we miss this, right? This is an important organism that's been studied a lot, but nobody ever found out about this, and this is kind of why. So homologs only exist in this one, the dark black here, this one fungal clade, and nowhere else in the genome. So there's no homology anywhere else in the genome. And so yeah, this possibly represents a new DNA binding motif. The structure doesn't appear to be homologous with anything else, really anything in the genome. And so yeah, the whole point of this is just, there are lots of cool targets that are still out there. Even in 2013, we're discovering proteins with completely new function, with completely new binding activities. So there's a lot of targets that are still out there waiting for people to find them. So here's one example of big data that we're going to pick on a little bit. So there was just a talk, I guess last week in IGB, we had a talk by one of you guys who runs PSI Biology, so Protein Structure Institute. So this is a multi-institution institute that's gotten $764 million over the last decade or so to ramp up high throughput crystallography to solve as many structures as you can of pretty much anything you can to learn things about them, basically. And that's about as vague as their mission statement really is. So they've deposited 2,900 unique structures since 2000. So that's almost $4 million per structure if you do the math, which isn't exactly the best success rate.
You can do it with the mass better.
76. Oh, did I move a decimal place? Oh yeah, sorry. But yeah, but it's pretty expensive. And the detractors of this project say that the problem is they're sort of picking out all the low-lying fruit that people might not necessarily be interested in. They're just stuff that's easy to crystallize, right? Is what's getting published. And so Greg Petsko is one of the more vocal opponents of this. He is a structural biologist, and he points out that the $60 million a year we spend on this is essentially 100 new PIs that don't get an R1, so they don't get tenure and they're out of science. And what he points out is perhaps more important is like solving structures for a good structural biologist. The solving structure isn't actually the bottleneck, it's actually the biochemical analysis chemistry. So one of the things he highlights is there's a couple big problems that crystallography can't solve that I think we can actually solve. So one of them is crystallographic conditions influence the oligomerization state. So the oligomer state you see in the crystal does not always correlate with the actual oligomerization state in vivo, and simple is one tool that we can use to actually tell us this. Another really great example, everybody likes to talk about Gleevec and how that's the future of modern medicine. When Gleevec was discovered, there was only one crystal structure, the BCR-ABL kinase, and it was the conformation that Gleevec does not bind to. Crystal structure was completely useless in discovering that drug and figuring out how it works. Also, membrane proteins are hard to crystallize, and I think we can do that pretty well. It's simple as well. So there's a need for rapid and more functional analysis of proteins of interest. So we need to move from taking snapshots of proteins and really studying how they work and how they move in a complex cellular environment. So this is just for people going back later. There's a lot of big data databases for DNA, RNA, a couple for protein. So I just want to highlight there's a protein localization database, fluorescence localization images for tons and tons of proteins. Yeast-2 hybrid is the best way to do protein-protein interactions. So there's huge databases already out there. But really, again, we want to know what these do in a cellular context. So here's the future. The future is robots. The future is supercomputers. The future is high-throughput cell culture done by robots. And the point is, that's not the future. All of this exists. On the shelf today, you can buy everything I've drawn in this slide. You can buy a robot to do all your cell culture, viral cell culture in a BL2 environment. You can get a cluster for cheap. You can get microfluidics that interface with an autofocusing, slide-moving microscope to completely automate microfluidic delivery of tens and hundreds of thousands of individual proteins into a microfluidic chamber that we can put on top of an injective turret we already have. So by the end of the decade, I predict that TJ will have cleared out the field. It'll have solved all the functions of all the proteins we care about, and there will be no use for postdocs anymore. So discovery methods, I'll go through real quick here. So ChIP-seq is a good way to look at pathways, and that's what we learned from the example in the beginning. We've done some examples using ghost limbs. So ghost limbs are in silico annotations based on sequence. So there's whole lists you can look up on the internet. Based on the sequence, they'll give you a list of proteins that presumably operate in a certain pathway. And then we can clone them and study them. So how do you get the protein in your hands? There's a lot of ways. You can call somebody and ask for it, and they may or may not send it to you. Gene synthesis is the future. And there's several libraries that you can buy on the internet. So pretty much any gene you can think of, we can get in a matter of days to weeks. There's a lot of tag protein libraries that have already been made in the literature, and all these libraries are now commercially available. Any organism you want, pretty much, E. coli, yeast, so 3 cloning strategies I want to go through real quick. Some of you have heard about these, and so I just want to throw a slide up for each. So gateway cloning is what we've been using in the lab, and so it's a recombinational technique that uses the lambda-phage machinery to pop a piece of DNA in and out inside of these ATT sites, which are recognized by the lytic and lysogenic pathway proteins from lambda-phage. So that's a way to not do a ligation, but you can pop a piece of DNA into an expression vector. So we have a library of these that we'll talk about in a second. The other cool new way is Gibson assembly. So this was invented by a guy in Craig Ventnor's Institute. Basically what it does is you take a vector backbone and you have to have pieces of DNA that are in your vector that are homologous. So this orange region overlaps with this orange region. These gray regions are homologous, and these blue regions homologous with this. And then you feed in a 5' exonuclease that generates sticky ends. So these sticky ends can then hybridize also in the mixture of the polymerase to fill in the gap, and there's a ligase to clean up the nicks. And now you've got a circularized plasmid without technically having to do a ligation. And Craig Ventnor's used this to make kilobase genomes completely available. Okay, so the last one is ligation independent cloning, also using an excisionase, so you do a PCR, and then you use the excisionase to chew back, and again you generate a long sticky overhang. That sticky overhang is long enough that you don't actually have to do a ligation, you can just transform it, and then the cell will actually stitch those together. So we did this using our HORF library, and in 1 week from knowing about a protein, we had a Western blot confirming that we were actually able to express this protein in human cells. And so the point is we have a library of this, over 12,000 human genes. So some of you already talked to me about this, but it's here. If you're interested in human genes, chances are we probably have some of your proteins. So I'm running out of time, so I'm just going to pitch. Obviously, the most straightforward thing we can do is a high-throughput screen for proteins that bind to DNA or translocate on DNA or have any activity that we can read out in a traditional fluorescence microscope in our lab. So by tagging the protein, you can immobilize the protein and float in a battery of DNA substrates rapidly, and again, this can all be automated. So in the course of hours, you can analyze dozens of proteins in a serial manner. And really what we need is downstream MATLAB automated, semi-automated analysis of all the movies that you generate to pick out specific phenotypes and fluorescence that mean something to us as biophysicists. So I'm running out of time. Okay, so I'm just going to rip through some possible experiments to think about. The point is essentially, any protein you want to look at, we can look at. So you can do sort of the standard experiments in our lab. Anything for storm imaging, hard to express proteins in E. coli. Now we have an expression system working in the lab to express anything we want in human cells, hex cells. So we can basically have any protein we want as a bait protein to fish out molecular complexes. So if you have PARP1, which Sui is interested in, so that should bind to histones, we can transform a tagged PARP1 into a cell to fish out whatever it is that you're looking for. So one nice thing we can also do with this is a fluorescent reporter cell line. Okay, fluorescent reporter cell lines where you can knock in an additional copy, or in this case for a reporter cell line, instead of knocking in the gene, you can knock in a plasmid with a promoter for that gene and a fluorophore reporter. So for viral work, you can envision essentially being able to light up a subpopulation of the cells that are affected by the virus in a different way by virtue of activation of the promoter. So I have lots more experiments, but I'm running out of time today, so I'll stop. Thanks.
All right, questions?
Well,
Yes, Alex.
just play a little devil's advocate on the automation aspect of it. From when I see everything's kind of separated already, the kind of commercial availability of it, it's not all one unit together. So what do you think is the kind of the work or the requirement is to actually get these working together? Like you're not going to just plug and play with that. What's the kind of the human capital input needed to make these work together?
So I mean, it really depends on what level you want to be working on. To be working on the level of dozens or hundreds of genes, we pretty much have everything in-house we need to clone and express already. Essentially, in a 96-well plate, if you assume you have a translation level of like 100 copies per cell, you can grow about 10,000 to 20,000 cells in a 96-well plate. That's enough protein to do a simple experiment. So you don't need a lot of protein, and really it's just a way to get those cells into a microfluidic chamber. And really, I mean, the hardest part is automating the microscope, but again, that's stuff that can be written in LabVIEW. It's stuff that you can buy off the shelf. So getting up past that, hundreds to thousands, now you're talking about robotics just to physically handle everything. But on the order of pathways we're interested in, I think we're already there.
All right, thanks. So next we have France, who is from France. She's a visiting student in TJ's lab, and she will tell us about chaperones.